Okay, so let me give you an example here. The case study here is a rare disease called aromatic L-amino acid decarboxylase deficiency. This is a very rare disorder of profound developmental failure. This is a child on the right, who I'll show you in a minute. More detail on seizures, lifelong care. The severe forms have a really catastrophic course and die before their 10th birthday. Though it is rare, it's certainly underdiagnosed and could have a prevalence of as high globally of 5,000 or 6,000 patients. It's certainly undoubtedly diagnosed different times, including cerebral palsy, seizure disorders, various things. So why did we take this one on? Well, because again, we're trying to develop general principles. So it turns out that the gene involved here is very well known. It's in the dopamine, norepinephrine, catecholamine synthesis pathway. So it's very well characterized. There is a PET ligand, a positron emission technology looking for dopamine synthesis in the putamen, which is a part of the brain that's particularly affected in this disorder. So one has a good biomarker to assess whether the gene therapy is working. To identify patients for clinical trials is quite straightforward. Monitoring the therapy is quite straightforward. And so that's why we decided to take this on. A number of things had been taken care of, so then we can focus on the technical problems that I mentioned. So I'm not going to go through all of these data, of course, but I want you to give you a sense of how this was worked. So this was a collaboration, again, as NCAT's projects always are, between the National University in Taiwan and a startup company called Agilis Biotherapeutics. The intervention here is a single dose, so-called AAV, adeno-associated vector, carrying human AADC gene, that's what that means, into the brain, the putamen, which is this part deep in the brain here. The challenges were a number. There's an ultra-rare disease, as we needed to develop a mechanism for stereotactic delivery in infants. There were regulatory issues, but what was particularly promising is that a number of patients, 18 actually, had received gene therapy in Taiwan with some remarkable clinical responses, which I'll show you in a second. So this collaboration started about a year and a half ago, to create GMP-grade manufacturing material, do all the GLP animal work, biodistribution and tox, do the patient finding, epidemiology, some natural history, and working on device development for delivery to infant brains. The good news, and why I'm telling you about this at this point, is that there was a very important regulatory milestone achieved this summer, an end of phase 2 meeting during the summer with our FDA colleagues, where they decided that no additional clinical studies would be needed in order to file a BLA next year. So this is currently the most advanced CNS gene therapy in development, and we think if this succeeds, it will not only establish a mechanism by which others can follow, but will now establish a regulatory mechanism that others can follow as well. So I want to show you this because we talk a lot about hypotheticals and general problems, et cetera, but we around here, I know, like all of you, think every day about people who might benefit from this technology. So I wanted to show you this. So this is the child, the picture of which I showed you before, and this is her when she's 4 years old, and her mom is showing her a ball and string, and you see she's really minimally responsive, reacts a little bit, but doesn't move spontaneously, is really minimally reactive. So she got the gene therapy at this point. This is her a year later. Now, it is formally possible that this is a spontaneous remission. Certainly formally possible, never been seen. Would stretch the imagination, but this is what clinical trials are for. And then A year later, this is her playing with her mom. Now, if all I did was to show you this, you would be hard-pressed to say that this kid has anything wrong with him. This is the same kid who 2 years earlier was sitting in the high chair. So this is incredible, just incredible. And why we decided if we can make this happen, what a huge advance this would be, this approval of this gene therapy. Okay, so we've also spent a lot of time asking, why did this work? The project, I mean. Why did the project work? It's something that you will hear around here all the time, but it bears reiteration because it is so unusual, I'm afraid, still. That is, there are clear strengths of a platform, of an experienced team with diverse expertise. This usually are public-private partnerships with patient advocacy groups deeply involved. We work almost exclusively on truly unmet medical needs, but we're starting out with well-understood diseases with biomarkers in order to get the technologies worked out, with learnings that can be applied to more difficult targets. So I want to emphasize that, again, the key success factors at the bottom are the team, the patient-focused and involved disease understanding and program, and the public-private partnerships have really been key.